This is the start of the lecture series for IA guidelines. So this is designed to help students understand the overall process of the IA and also provide detailed accounts of what individual sections in the IA report would look like. So then they can follow the lectures and understand what to put in their IA draft. So IA is self-directed investigation. So students will receive minimal help from their teachers, and they will have to choose their own topic of investigation and carry out an experiment and write a lab report on it. According to the IB guidelines, it should take about 10 to 15 minutes of effort, including the lab hours. However, for a lot of students who are trying to get 6 or 7, those high scores usually require more hours because it is a very extensive process and also it takes a lot of effort. When it comes to the lab report itself, there is a word limit of 3,000 words, so students should keep their report under that limit. Anything that goes beyond that limit, the examiners and the school teachers might not read. So if their last sections of paragraphs might suffer from those. And lastly, this is not just collect data and present it in a pretty graphs, but more so it requires a lot of critical thinking and analysis and evaluation from the students. So the IA will take up in total 20% of the student's IB grade, the final grade. And when it comes to the IA rubric itself, it has 4 criteria, A, B, C, D. And so A has to do with the experimental design aspects, B has to do with data analysis, and C is about the conclusion, and D is on evaluation. And they each take up 6 points, so then in total it will be 24 points worth of mark. The general process of IA, now beware that this might differ depending on school to school. Some schools do it in smaller chunks. Other schools just give like a huge IA project month where students are required to finish it within a month or a month and a half or so time frame. However, it usually starts with the topic selection. So the students will be asked to come up with a topic that they're interested in. And they will often have to send in either some kind of form or a proposal document to the school teacher. So then the school teacher can approve the topic and make sure all of the materials can be prepared by the school laboratory. And once this topic approval and the research topic decision is done, then the student will write out the background and the method sections of their IAs, and they will actually go into the experimental phase. Now, a lot of the times experiments don't really go as intended, and a lot of scientific experiments fail, especially in biology. If you're dealing with living specimens, sometimes they just won't cooperate. So if that's the case, then they will have to do some troubleshooting to maybe fix up their conditions or the duration of the experiment and so on. So once all of this troubleshooting is done, they should update their method to the final version and then complete their data collection. Once that's done, they will complete the first draft and then submit their first draft to the school teacher for a feedback. And then based on that feedback, they will have a moment to, usually a week or 2 or so of time, to edit their first draft and then submit their final draft. The IB doesn't have strict rules on the organization of the lab report itself. However, most lab reports will follow a certain order. So they will start with a title page, given, and then the introduction, the methodology section, then the results presentation, and then they will conclude the results, and evaluate, and then lastly provide the works cited or bibliography sections. But optionally, if students have very lengthy raw data or additional documentations, then they can add those into an appendix section. The title page should, of course, first of all, state the topic of investigation very clearly. But in addition, the candidate's IB number should go in there as well as the final word count. The introduction section should typically begin with a research question clearly stated at the beginning of the document. followed by any scientific concepts that has to be explained. So these are any scientific details that are not typically covered in the biology syllabus itself, but will be necessary for any readers to really understand the topic of investigation. And then typically the students will put the variables section next to it after the background information. So this includes the independent variable, dependent variable, and the controlled variables. And this can be provided in paragraphs format or tables. Usually for control variables, it's much easier to put it into table format because then students can easily list the 5 control variables and then put justification side by side. And then lastly, it is optional. However, it is beneficial to have hypothesis section. It should be just one very short paragraph. However, this would benefit the students for writing the conclusion section because they have something to compare to. The methodology section typically begins with a list of materials. So any materials, equipment, reagents, living specimens that students use in their lab, they should specify and also indicate the quantity of how much they were using. In addition, if they were using any equipment, measurement tools, then they should specify the uncertainty that goes along with it. Now, this is optional, but there are preliminary testing usually sessions in the beginning of the data collection. And this is a lot of troubleshooting and finalizing the details of the measurement conditions. So for example, if the student initially tested for say 5 minutes, but then it wasn't enough to see some kind of observable change, so they had to extend each trial to 10 minutes, then they can write a short paragraph on what were the results of the preliminary testing and how they adjusted the final procedure according to those results. And then they should give the final step-by-step, the finalized procedure, which this is not an outline of the experiment. This needs to be a detailed account where if someone else is to read the procedure and replicate the student's lab, then they should be able to follow the experiment without any questions. And last but not least, for biology, students must give risk assessments, which includes the safety, environmental, and the ethical considerations, and especially ethical considerations if the students are using living specimens, such as plants, bacteria, or some kind of tissues. In the results section, the students typically start by presenting their raw data. This includes both the quantitative and the qualitative observations. The quantitative data typically is organized into a raw data table. However, depending on the experiment, if it's collection of data, as in logger pro screens or graphs that are screenshotted, then the students can put this in the appendix because it will be very lengthy. But after presenting that typical raw data, the student then needs to go on and provide process data. So usually this involves calculating the average and standard deviation for biology, and then providing some kind of final visual representation, such as a histogram, scatter graphs, or pie charts. It really depends on the topic of investigation. And last but not least, in order to strengthen the results, then a lot of students will resort to some sort of statistical analysis to make sure if their data, collected data, is significant in terms of statistics. So this involves things like t-tests and ANOVA tests. In the conclusion section, typically it's a great idea to start by reiterating the aim of the investigation, so paraphrasing the research question. Then clearly state whether the data supports or to what extent the research question is answered by the data collected. And if the student has hypothesis section, then this is a great place to answer. Was the hypothesis supported or not supported by the results? And if so, to what extent was it supported? So how reliable is that? And then in addition to just summarizing the data, the students needs to link that result to scientific context. So in addition to just simply saying that there is an increasing trend, then they would have to connect that to the scientific theory of why is it correct to see an increasing trend in the data. So this is referred to as a scientific context by the IB. And then lastly, if there are any literature, any other scientific articles to compare to, then this is a great place to put in, and then bring in some other excerpts of data or quotations from other outside scientific articles to add further supportive details. In the evaluation section, first of all, the students should start by evaluating the strengths and the weaknesses. And so this both evaluates the method itself and the data collected. So what were the strengths and the weaknesses of the method of the experiment, and also what were things that went wrong? So were there any anomalies? Were there any conditions that students initially didn't consider, but it actually affected the data collection and things like that? And also with any identified weakness, they must supplement with appropriate improvements. So if they were to repeat this experiment one more time, then what are some realistic improvements that they can make to the experimental conditions? So then they will minimize the errors. And then last but not least, it is a good idea to include a final paragraph which explains any limitations. So to what extent did their research question was answered by the experiment, and was there any limitations to the scope of the experiment itself? And if there was, then they would add a paragraph on what further investigations could be done. In the reference section, the students should follow the formatting style of works cited that usually their school uses. So this could be things like MLA formatting or APA style, whatever the teacher specifies. Usually they are specified by the school teacher. And so they should have the full works cited section, but also they must double check that they have the correct in-text citations within their draft. Optionally, again, as I said previously, if their raw data is too lengthy or if they have any other supplementary documents such as surveys or consent forms from their peers, and this often happens when students decide to do physiology related IAs where they make their friends like run treadmills or so. And so any of those supplementary documents would go into the appendix section because otherwise the main draft will be too lengthy. Word count is probably what a lot of students get confused because there are detailed rules about what to include and what not to include in the word count. So any writing typically will be counted as word count. However, any numbers in tables and graphs are not included in the word count, as well as their section titles and graph and table titles. Additionally, anything that is outside of the main text, so such as any in-text citations that go in the parentheses, would not be counted as part of the word count. As the last section of this introductory lesson, I would like to give some guideline on how to use AI in a healthy way, because in modern days, AI use in studies and academics is sort of inevitable. A lot of students will use them, and the IB actually does not prohibit students from using AI tools. However, simultaneously, IB is very strict on the academic integrity rules, so I would like to introduce how to use AI tools such as ChatGPT or Gemini in a way that does not go against the integrated rules of IB. So first of all, any AI-generated text must be cited in the works cited, including the prompt itself. Now, something that I would highly recommend and emphasize in students is that, especially when it comes to ChatGPT, the AI tends to lie quite often. So instead of using the information provided by the AI directly, use it as a starting point of your own research. So you would ask the AI to give some sources, some summary of the facts, and then the corresponding primary sources of scientific articles, and then actually go visit the primary articles and read them to use in your background research and to provide your scientific context in your IA. So let me show you how this demonstration of how an example AI use might look like. So for example, let's say I'm interested in investigating the effect of micronutrients on the rate of photosynthesis. So let's say I read into articles saying that the soil quality does matter a lot in the extent of photosynthesis, and I would like to investigate the details regarding that as my topic of investigation. Now, because I'm not a plant, I don't really know what kind of minerals might be beneficial for plant growth. So let's say I go to ChatGPT, I open up a new browser and ask, give me some list of micronutrients that are required for plants to do photosynthesis. And in that prompt, I would add to make sure to include the citations for the sources. So then the ChatGPT will give me some response with the lists of minerals, say iron, magnesium, calcium. Let's say I get like 5 or 6 different lists. I would briefly read the individual paragraphs and see the general summary of how those minerals are important in plant physiology and photosynthesis, and say that iron looks promising. Then I would ask follow-up questions such as how does iron affect photosynthesis, and include the primary sources. I might say give me 3 specific scientific articles, and if I want more recent studies, then I might even specify between which years. Then after the ChatGPT gives me the response, I would go visit the primary articles and read those. In addition to ChatGPT, usually Gemini might do a better job in giving primary sources, so maybe for that follow-up question, use Gemini. Once I have the topic itself, let's say I'm trying to investigate the effect of iron levels on the rate of photosynthesis, then next I need to decide on which plant am I going to test with. And so again, I'm not a botanical person, so I don't really know many plant species. So say I'm going to go back to the ChatGPT and ask, what are some good model organisms, model plant species to use to test photosynthesis? And I might ask the ChatGPT to be a little bit more specific and give me 3 terrestrial plants versus 3 aquatic plants, because they will, of course, change how the lab setup will look like. And then I would read through the details and then select one final species for my topic of investigation. Before we wrap up this section, this introductory lecture or section to the IA journey, I would like to give some tips for keeping your primary sources. So the IA procedure will typically require lots and lots of research, and sometimes it is so easy to lose where you found certain details, and later if you want to quote that or paraphrase that, it is important to keep all of those sources. And for that purpose, I would like to suggest using tools such as Zotero, or additionally other schools might use tools like Noodle Tools, and there are lots and lots of those different websites. But please keep all of your research sources together, maybe saved in a folder inside Zotero or Noodle Tools, so then you can easily find those sources later when you're actually writing the entire draft.